led to genomics assisted breeding, because we know the urgency is there to produce speedily the food production. So we can't go by the conventional methods because it takes a lot of time. So we can accelerate the conventional breeding using precise and efficient genomic tools. So recently there is a variety called Geletu, which is a product of genomic assisted breeding in chickpea and has been released in Ethiopia. Is a high yielding variety. We have another variety which is very recently released in India in chickpea, that is drought tolerant and fusarium wilt resistant varieties. Similarly, in case of groundnut, we have high oleic groundnut varieties which have been developed and ready to release. Similarly, foliar disease resistance groundnut lines are also in the ECRIP trials. So these are one area where we are trying to address malnutrition by having high yielding varieties, but at the same time we also have to consider malnutrition from a different perspective. Why a malnourished child, even though they are given adequate food, they still fail to grow? Why? Because they are unable to assimilate the food. Some of you might not agree to me, but there are 40 trillion bacteria in your gut. They are doing your job. They are actually absorbing the nutrients for you. So there are good bacteria and bad bacteria. So what we are trying to do is, the malnutrition is a vicious cycle. Let us understand it. So when a poor gut microbiome is there, it will fail to absorb the nutrient in your body, leading you to an undernourished and anemic state. When these children, adolescent girls, grow up, become a mother, as undernourished mother, give birth to an undernourished child. So this vicious cycle goes on and on because the mother is transmitting the poor gut microbiome to the child, the next generation. So what we have to do is we have to chip in. So what we are trying to do at ICRISAT, with partnership with ICMR, National Institute of Nutrition, what we are trying to do is we are trying to profile the gut microbiome of anemic girls, like what is the status of the gut microbiome by metagenome sequencing, and then we are feeding them a cohort will be given government recommended IFA tablets. They are the iron folic acid tablet, and we are profiling them again. Similarly, we believe in that the dietary intervention is more effective because it is correcting the gut. So we are trying to formulate, we already have formulated a peanut pearl millet based bar which is being given to these girls, and we are seeing how they are correcting the gut and how much they are being absorbed and their status of iron status and their overall health, how they are improved. So this is what we are trying to do, and this project is already, we have started in two phases. This project is named as FIFA Girls, that is iron for adolescent girls, with a partnership with ICRISAT and NIN. A second study we are doing, which is for studying the undernutrition in children under 5. So this is a multi-centric project, and this is already being approved by ICMR for funds. And then what we are trying to do is we are looking at the gut microbiome profile of children under 5, because even they are being fed with nutritious food, they are unable to assimilate it. So what is the profile of that, and how we can correct the gut by giving the finger millet based, that is ICRISAT mandate crops based food. So in this we are doing countrywide, and there are 4 centers for this. National Institute of Nutrition, we have Trivandrum Medical College, Kerala, Society for Health and Democratic Surveillance, West Bengal, and also ICRISAT's Agribusiness and Innovation Platform is also involved. So now it's about the gut microbiome. We are also studying the soil microbiome for crop productivity and environmental sustainability, because we believe that after Green Revolution, the amount of fertilizers what we are using and that is going into the water bodies and polluting. So can we use the soil microbiome for supplementing the nutrition to the plant? So what we are trying to do is, soil is the main link to the micronutrient contents of food production, and the root microbiome accounts for more than 60% of the total microbial population and activities in the agricultural soil. So these soil microbiome can actually give the plant an immunity from a pathogenic outburst. Also, the soil microbiome induces resistance to the above ground insects in plants. Similarly, soil microbiome can impact traits like influencing the timing of flowering, etc. So at ICRISAT, what we did is we collaborated with IARE Delhi, and we have set up an experiment where we have a panel of chickpea genotypes which will be actually grown in these two geographically distinct locations, and then we profile the soil microbiome of both these locations. So what is the thing happening is, certain genotypes. So this panel of chickpea genotypes were actually released during last 7 to 8 decades. So before Green Revolution, after Green Revolution, some of the genotypes and some of the varieties, they have the power, they have the potential to use the soil microbiome in a better way to have a better crop productivity. So we are studying that. So what we are trying to do is, we are trying to link the composition, diversity and the abundance of a soil microbiome composition to the function and associating it with the improved crop productivity traits. And we are defining a functional microbiome which can further be used as a symbiotic community, which is called a SymCom, and this SymCom can be introduced into the plant, into the soil in the form of seed coatings or in some other way like soil transfer. So there are studies in foxtail, millet and all. They have clearly shown that soil microbiome, there is a marker O2s which are actually responsible for positive yield in the plants. So we can actually go for a designer microbiome which can be introduced in the soil. I already mentioned that. So the take-home message from this is, agriculture is key for a sustainable solution towards healthier diet and reducing a burden of malnutrition. Dietary diversity is vital. Genomic assisted breeding can speed up the to speed up the crop productivity, hence the farmers' profitability. Gut microbiome and soil microbiome are some emerging areas which has to be explored, which can improve agriculture and human health. Human gut microbiome is not only critical for nutritional absorption but also for the maintaining overall health. Employing soil microbiome can reduce the dependency on the fertilizer for a greater environment sustainability. That's it for now. Thank you.
Thank you, Dr. Lekha.